Welcome to Grade 9 Exploration New Curriculum, Chapter 2, Cell, the Building Block of Life, Part 2. Today we are going to learn about cell membrane, structure and function, especially fluid mosaic model, concepts of osmosis and diffusion. We will conclude with cell wall in today's lesson.
We've measured the cell, but what defines its boundary? Let's zoom into the universal feature of every cell, the membrane. Beneath the microscope, this thin line is actually a complex bilayer of phospholipids, acting as life's first protective barrier. This membrane isn't a rigid wall, it's strictly fluid. These lipid molecules constantly glide side to side. This is the fluid part. These molecules aren't stuck, they shift and flow like a sea, allowing the cell to be flexible and resilient. And here is the mosaic. Large proteins float within the lipids, serving as channels, sensors, and structural supports for the cell.
Remember our onion cell at 200 micrometers? The membrane is only 7 to 10 nanometers thick. That's nearly 20,000 times thinner.
In our unit conversion, 1 micrometer equals 1,000 nanometers. Understanding this scale is the key to mastering cellular biology. Scientists discovered this structure by carefully peeling back the layers using advanced microscopy, proving the mosaic theory. From the scale of a ruler to the dance of molecules, you are now seeing the world beyond the naked eye. Keep exploring. How do plants drink water without mouths? The secret lies in osmosis, the elegant diffusion of water across a cell's membrane. Let's see it in action with a potato osmometer. First, peel a raw potato and carefully hollow out the center. We carefully pour the heavy sugar solution into the potato cup. A metal pin is then inserted precisely at the
Leave it for a few hours and watch. The water level inside rises from the bottom up. But how did the pure water get through the solid potato? The pure water outside is a hypotonic solution. Because it has more water than the inside, water rushes in, swelling the cells. If we reversed it and placed the cell in thick salt water, a hypertonic solution, water would leave the cell, causing it to shrink.
And in an isotonic solution, the concentrations are perfectly matched. Water moves in and out evenly, keeping the cell completely stable.
Our potato acted as the perfect gatekeeper, balancing the flow of life. Try this beautiful experiment at home and watch osmosis in action.
Start by peeling a thin layer of onion skin. Place it on a slide and add a drop of safranin to reveal its remarkably organized structure.
Next, gently scrape the inner lining of your cheek. Spread the cells on a slide and add methylene blue to bring these transparent animal cells in.
Under the lens, onion cells look like organized bricks with rigid walls, while cheek cells appear irregular and loosely arranged, distinctly different architectures. What happens when the external environment changes? We introduce a concentrated 20% sugar solution to both samples and observe the results after 30 minutes. Observe the plant cells. The inner contents shrink dramatically, but the rigid cell walls hold their shape, a fascinating process known as plasmolysis.
In contrast, animal cells lacking cell walls shrink entirely. This highlight underscores the critical role of the cell wall in protecting and supporting plant life.
Now, let's learn about cell wall.
Unlike animals, plants cannot move to escape the elements. To withstand wind and rain, their cells feature a rigid outer shield, the cell wall. Primarily built from tough fibers called cellulose, this wall keeps plants upright. Yet, it remains selectively permeable. Microorganisms like fungi and bacteria also rely on strong cell walls for essential protection and structural support. Animal cells don't have cell wall at all. This lack of a rigid boundary provides the cellular flexibility. Let's pause and ponder. Why do stationary plants need a rigid cell wall, while moving animals do not? Plants can't hide from storms, so rigid walls protect them. Animal cells stay wall-free to bend and move.
Question 2. What if a plant's rigid cell wall became as soft and flexible as its inner membrane? Disaster! Without a rigid wall to handle water pressure, the cells would burst, and the whole plant would collapse.
Finally, Think back to our potato experiment. Why must we cut equal pieces and weigh them before starting?
It's for scientific accuracy. A precise baseline lets us calculate the exact amount of water gained or lost.